Hi, this is Mr. Matthews, and I'm going to go over just a little bit about what the cell membrane and diffusion lab was about, and maybe what you should have got. For those that were absent, this is a good way to collect your data, and for those in first period especially who uh, weren't able to finish and collect their data, we can use the data that's in this presentation. So just keep in mind that you should have the access to the lab write-up from Google Classroom. Just click on the link. You don't need to save it. It automatically saves. We're just going to fill it out right here. And this is a smaller version of the Science Fair template. So you'll have some class time to fill that out. But what we're looking at is now that we have an idea of what the structure is like, we want to start talking about the movement. We want to talk about the term selectively permeable. The cell membrane is considered a selectively permeable membrane, means that it allows some things in and others not. And we want to look at the scenarios that take place in our bodies that allow things to come in and come out. And that's what diffusion really is all about, the movement of molecules from a higher concentration to a lower concentration. So we're going to play around with what's called dialysis tubing to figure that out. So I want to draw a couple of pictures here. There's two parts to this lab, and the first lab, part A, was done by half the students. And what the whole purpose of this part was to really determine if there was an increase in mass in each one of the scenarios. And what I mean by that is that we filled this dialysis tube, and then the dialysis tubing is meant to simulate what the cell membrane is like. It's very much like a model membrane. And we filled this dialysis tube with 20% sucrose. We filled 3 of them because we're going to put this dialysis tube, which is supposed to simulate the cell membrane, with 20% sucrose in 3 different scenarios. We have cup 1, we have cup 2, and we have cup 3. In cup 1, we had just water, 0% solution. We plopped in a dialysis tube filled with sucrose, and we let it sit. Okay. Cup 2, we had a 20% sucrose solution, and we plopped that dialysis tube in, and we let it sit. In cup 3, we had a 40% sucrose solution, and we plopped another dialysis tube in there filled with 20% sucrose, and we let it sit. We let it sit for 30 minutes. Now, prior to plopping them in there and letting them sit, we actually took an initial mass in grams. And after we let it sit, about every 5 minutes, students pulled the bags out and wrote, visually, did they see the bag getting bigger, smaller? Did they feel like it was gaining mass or losing mass? And then, again, every 5 minutes up until 30 minutes, and then they actually recorded the mass. So when we did this experiment, and what other groups did, and before I give you that detail, I want you to think about what you think is going to happen. Do you think one of the baggies is going to gain or lose mass? And when you think to yourself what might happen, that's what the hypothesis is all about. So before you write that down, you might want to consider writing a hypothesis. Here we have a very basic hypothesis. You can choose to use this, you can choose to use another one. Either way, you need to write a hypothesis down before you actually look at the data. So this one says if a dialysis tube is placed in a 40% solution, it'll lose mass. And if we look at our picture, they're looking at this one right here. If this dialysis tube filled with 20% sucrose is put in a 40% solution, the mass is going to decrease. Okay, and you don't have to be right. This is just a guess. So when we look at the data here, and this is what we collected, after 5 minutes, everything stayed the same. After 10 minutes, it looked like the 40% was getting smaller. After 15 minutes, it looks like pretty much the sugar solutions were getting smaller, and the same at 20 minutes. So after 20 minutes, let me correct that spelling. After 20 minutes was up, we came and looked at the data again. So the 0% solution, if we were to weigh it out, we started with 23, ended with 25. And these are grams. Make sure you put grams in there. G for grams, 25 grams. In the 20%, it looks like it stayed the same. 40% stayed the same. So what we're trying to identify whether or not this model membrane is semi-permeable, and potentially what scenarios would cause water to move or sugar to move in and out of the cell. So based on these observations, do you have definitive quantitative and qualitative evidence that there was any movement? Again, we're looking at the word selectively permeable. Is the cell membrane, or can we prove the cell membrane is selectively permeable? So look at this evidence and think about it as we move forward. Part B was a little more complicated. Part B, I'm going to go on to a next page. So part B, we're going to have a cup here, and we're going to fill a cup with just regular water, and then we're going to put a dialysis tube, and I'm going to say half of the tube is sugar, the dialysis tube, half of it has sugar, and half of it has starch. Okay, sugar and starch, the two of them together. So what you're looking at, what you're looking for, okay, we let this sit for 20 minutes, 15 to 20 minutes. We want to see if any of this sugar and starch actually diffused out of the bag into the surrounding water. That's what we're looking for. So we're going to have to test the water. So we pull some of the water out, and we test the water on the outside for each of these. We want to test it for sugar and starch. All right, 2 tests involved. Before we can do that, we have to know what a positive sugar test is. We also have to know what a positive starch test is, what it looks like. Okay, so prior to doing even this setup, these groups actually had some tests to do, some prior knowledge tests. So in a starch test, let's say I have this test tube, and I have this test tube. Let's say that in test tube A has water. In test tube B, here's test tube A. Test tube B has starch. And we fill the test tube up with water and starch. And test tube A, when I just add in the indicator, it turns yellow. Well, what if in test tube B, I add in the indicator, it turns black or purple? Okay, so the whole point is that if you are running a test for starch and you get black or purple, it means that starch is present. So going back to this page, if I were to pull some of this water out right here, okay, if I were to pull some of that water out and do a starch test, I would be looking for the solution to turn black. For glucose, I've given you the glucose solution. I have given you, right, I have given you the glucose test. So what this means, if you are doing a glucose test and you wanted to see if glucose was present, and you were looking for an orange or brown color in the test tube. If not, then it means no glucose is present. Pretty much blue, blue, and even clear. So again, if glucose was present in the water, if you wanted a positive glucose test, then you're looking for a solution that's orange or brown. So back to your data table, again, the starch test, it was just water, it was red or orange, no starch is present. When you added starch in the indicator, it turned purple or black, that means starch is present. for the glucose test. If it turns orange or brown when you're using the glucose indicator, it means glucose is present. So that's what you should get. So when students actually pulled the water out and did the sugar and the starch test, here's what they found. They found that when they did their starch test, it turned red and orange. And again, going back to what we should have gotten, if it turned red and orange, it means no starch was present. So that means that no starch was able to get out of the bag. When they did their glucose test, it turned light blue. Well, light blue, if we look at our, anytime we have blue, 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 it means that no glucose is present. So that also means that the glucose couldn't get out of the bag. It still stayed in the bag. It didn't diffuse through. The last test we did was an iodine test. So not only did we see if sugar and starch could get out of the bag, we also put some iodine in the bag. And we wanted to know if iodine could diffuse through the bag. We wanted it to diffuse through the bag and make that black, okay? And here is what we found when we did that. Right there. This is what we found. We put the bag, we added the iodine to the water, and the iodine diffused through the bag, making the inside part of the bag black. So if we go to our data table, we have when we added the iodine to the outside part of the membrane, it diffused in, meaning that iodine can enter the bag or the cell membrane. So looking at all of these results in general, for Part B especially, the whole idea is that this was a model membrane. The dialysis tubing was a model membrane. And in order to be a model membrane, you have to be selectively permeable. And based on these results, you need to think to yourself, was this a selectively permeable membrane? Was everything able to get through? Well, based on our results, the only thing that was able to get through the bag was the iodine. Everything else was not. If starch and glucose wanted to get through the bag, they would need either larger holes or a tunnel like a transport protein, much like you needed to put a piece of string on your bubble in the bubble maker lab. So once you have this data entered, go ahead and answer the analysis and discussion questions.